Hello, my name is Ed Moore. It's a pleasure to join you in this workshop. Thanks to Costas and the organizing committee for the invitation. I am at the University of Gothenburg and the clinical microbiology lab of the largest hospital in Sweden, where my research group is working on improving diagnostics of bacterial pathogens and antimicrobial resistance. And I'm the director and curator of the CCUG, which holds the largest collection of clinically relevant bacterial bacteria in Europe and perhaps the world. The research in my labs has been focused on DNA-based analyses of microorganisms since I was a PhD student. Since about 7 years, we have been employing genomics and proteomics for trying to improve diagnostics. Our work necessarily has included trying to understand taxonomy and systematics. It would be impossible to establish effective diagnostics for infectious diseases without a reliable taxonomy of microorganisms. I will talk about the importance of nomenclatural types to be strains rather than DNA sequence data. And this, I would say, controversy is connected to a vote last year of the ICSP to change the prokaryotic code, which regulates issues of naming prokaryotes. Because of this controversy, this workshop is considering an alternative taxonomy to the taxonomy regulated by the code. Taxonomy is relevant for various aspects of academic, medical, and biotechnology research, and in most aspects of microbiology. Even if microbiologists do not consider themselves to be taxonomists, most of them will be dependent on or apply some aspect of taxonomy in their work. Taxonomy is comprised of three components. Firstly, characterization has used two basic strategies: phenotyping and genotyping. Phenotyping is the traditional means for microbial characterization, while genotyping has been the method of choice in the last 30 years. Unfortunately, reliance on genotypic methods has resulted in a trend for a minimalist approach for characterization rather than comprehensive polyphasic approaches. Classification is the second component of taxonomy, which is the arrangement of bacteria into taxa on the basis of similarities or relationships, that is, the results of characterization. Bacterial classification, still to this day, follows the general scheme developed by Carl Linnaeus in the 18th century. Nomenclature is the third component of taxonomy. This is the assignment of names to the taxonomic units that have been characterized and classified. For maintaining the integrity of nomenclature, the International Code of Nomenclature of Prokaryotes, or the code, was developed. I point out that the only aspect of microbial taxonomy that the code deals with is nomenclature. The code does not consider issues of characterization or classification. The bacteriological code, or since 2008, the prokaryotic code, has been developed over many years. Attempts, even at the beginning of the 20th century, were made to try to codify a set of rules or a code to ensure stability in bacterial nomenclature. In 1947 and 1948, Robert Buchanan and colleagues established a bacteriological code of nomenclature for microbiologists. This was due to a split by the microbiologists away from the botanical code. This split was due in part to a disagreement between the microbiologists and botanists over pushing for depositive strains to serve as type material rather than relying on non-living samples or illustrations, as is common in botany. So there is a history of splits due to different philosophies in bacterial taxonomy. The first edition of the code in 1958 has undergone several iterations until we have the current edition, which was revised in 2008. And it will be revised again in the future. The next edition is expected to be published in 2022, which will address proposals for changes since 2008. The code functions based upon principles, rules, and recommendations. The principles form the basis of the code, and the rules and recommendations are derived from the principles. So here is where the problems begin for those who propose to use genome sequences as type material. In the code, the description for the type strain is given. That is, it is made up of living cultures of an organism which are descended from a strain designated as the nomenclatural type. And the characters of the type strain should be defined. The bacteriologists inserted the phrase type cultures in 1948 in their new code, but kept the wording of the botanical code, which refers to the use of non-living materials and illustrations, etc. And since January 2001, only strains may serve as nomenclatural type material. This was defined in Rule 18A in the 2008 edition of the code. Under Rule 30, until December 2000, a culture of the type strain of a proposed new species was only recommended to be deposited in one culture collection. After January 2001, a culture of the type strain of new species must be deposited in two collections in different countries. Why the rule change? Simply because of too many instances of the type strains not being available anywhere. Such a situation was not workable for at least two reasons. Firstly, for taxonomic research, the reference strains must be available to the scientific community for comparative analyses. Secondly, for any kind of research, the work must be repeatable. Why deposit into two culture collections in different countries? To reduce the probability that the most important strains used as a reference would be lost. That is, if one collection were to lose a type strain, it would still be available in another collection. Also, there were instances of researchers in some countries trying to control the distribution of strains from the collections in those countries. If the strains are deposited in collections in another country, it is more difficult to control the strains. Furthermore, evidence must be presented that the strains are available. So, since January 2001, collections have been providing CODs, or confirmation of deposit or certificate of deposit, to depositors that the strains are available. These are requirements by the code for valid publication in ICNP of new names of bacterial taxa. Valid publication provides standing in bacterial nomenclature. Valid publication does not validate the characterization, classification, or any other systematic work on bacterial taxa. In any case, the code allows for exceptions to the Rule 30. In January last year, the ICSP considered a proposal to change the code. The proposal change was to allow the use of complete or partial genome sequences as type. For amending the code, it is required that the proposals for changes should be published in IJSEM and then discussed for a period of 3 months. This will be done as an online discussion, open to members of the ICSP, but open also to any microbiologist. You can see the proposal along with the rules that would have to be amended. The new text to be proposed is underlined. The proposal would allow for complete or partial genome sequences to be accepted as type material, in addition to or instead of strains being the designated type material. When possible, the sequence should be of high quality draft or better. When a sequence is the type, it is recommended that samples of the DNA be deposited in 2 collections. The voting on the proposal would be among the ICSP and the Judicial Commission members. So for the online discussion, there were also publications arguing different points. Among these dueling publications were one from Costas, Ramon, and Rudy Aman, another from Ramon and Barney Whitman. Both of these argued for the proposals. A paper published by Jörg Overman and colleagues and by Magna Biscard, both of these argued against the proposals. And even after the discussion period and the voting was finished, Costas and Alison Murray published, with their colleagues, published papers in favor of the proposal. The 3-month discussion period for considering whole genome sequence data as type material resulted in a vigorous debate. 107 contributions from microbiologists around the world. So I organized the main issues of concern into categories, including sequencing concerns, taxonomic concerns, concerns for reproducibility and confirmation of research results, and comments on relevance. Here below, I will indicate a few of the points of concern. However, since the presentation is online, the participants can read the specific concerns that were raised during the discussion. Here are some comments regarding sequencing concerns. I will not read them. The presentation is online, so anyone can check the different comments. Here are comments regarding concerns about taxonomy with the change in the code. Here are comments regarding concerns for reproducibility of research and comments on relevance regarding adopting the change in the code. I would mention that the results of the vote were decisive, with 76% of voters rejecting the proposal to change the code about what would be accepted as type material. So, although the proposal was not accepted as a solution to the issues for validly publishing names of the uncultivated, I think that all microbiologists agree that there are problems that need to be addressed. I would pose the problems by differentiating between different kinds of uncultivated, as their issues are not necessarily equivalent. A first group of uncultivated are the hyperfastidious prokaryotes. These require specialized labs and experience to work with such bacteria. For those that are not able to be cultivated, genome sequence data could be archived, as well as DNA samples. But even if these strains could be cultivated and sent to culture collections, the collections would not be able to work with them in most cases. One collection, the DSM-Set in Germany, is probably the gold standard among culture collections. They provide services for analyzing microorganisms at all levels and are able to work with most prokaryotes. They have the personnel and, importantly, they have the expertise to work with most prokaryotes. Not many collections are able to do that. In our case, at the CCUG, we are 7 persons. Additionally, we are a collection focused on clinically relevant bacteria. We are not able to work with archaea or obligate symbionts, etc. Some years ago, I received a request to accept almost 200 strains of cyanobacteria. The professor was very put out that we could not accept the strains. A partial solution would be funding support for collections that would accept the hyperfastidious strains, work with the genome sequence data, and with DNA samples. A second group, taxa detected by SAGs. The issues may be somewhat similar to those of the hyperfastidious. Even for those that cannot be cultivated, genome sequence data could be archived, as well as DNA samples. A third group of the uncultivated, the MAGs, provide an extensive overview of biodiversity in a sample with tens of thousands of potential OTUs. Are all of these reliable estimates of taxa? Are they hypothetical species? How should these samples be archived for taxonomy purposes? There is also a problem in that if DNA samples are archived in a collection, the question is, how would the collection be able to capitalize on those samples? It is clear that microbiologists need a taxonomic system that is able to deal with the huge numbers of taxa among the uncultivated. This is an issue that culture collections have not faced before. What would be an alternative taxonomic system for the uncultivated? How would that look? With an estimated 10 to the 30 species of prokaryotes in the world, and with tens of thousands of OTUs discovered in each metagenomic study, is it realistic to depend upon a Latin-based binomial taxonomy? I would have proposed a numeric or alphanumeric barcoding system. Examples of such systems already exist. The coding behind the Latin names of NCBI taxonomy is a numerical system. However, before this workshop, I did not realize that a C code already existed. So I am looking forward to learning about that system. Alternatively, perhaps make the names longer and longer. Here you have an example of a new species of Myxococcus with the longest species name, none. This was published last year, I believe. So I thank especially these people that I've had nice discussions about taxonomy over the years. And I thank the contributions of the ICSP online discussion for the proposal on the use of genome sequence data as type material, as well as the ICSP Executive Board, and the Judicial Commission members. I acknowledge that comments presented in this talk are the ideas from many of these people which have been circulated in the discussions. Thank you for your attention.